Hello, this presentation is on scientific methods in biology. So the goal of all science is to objectively explain the processes in nature of the universe. Scientists aim to be unbiased in their observations. Please be aware that scientific knowledge is dynamic and constantly changing as we learn more and more information. This is what we call scientific method. So what are the steps in scientific method? And I know that many of you have been doing this for several years. We start off with an observation. This is what we see. It's a question that we ask, something we observe in nature, and we want to know more about it. Then we do research. We figure out, has someone already figured out that question? Next, we create a hypothesis. This allows us to answer that question. Then we design an experiment. This is the fun part we all enjoy doing. Next, we collect data from that experiment. From that data, we then analyze the results. Then we retest it. That's something that's going to be critical, is to test and test again. As scientists, our results need to be reproducible. Anyone from anywhere on the planet need to be able to do that same experiment and get the same results that you did, to ensure that what you're observing is in fact true. That is the steps in scientific method. So let's analyze these in a little more detail. The first one here is observations. These are things that we gather through our senses. As scientists, we are inquisitive about nature. We come up with lots of great questions. So an example of an observation might be noticing that many salamanders near a pond have curved and not straight tails. The next step is the hypothesis. The purpose of the hypothesis is to suggest a solution to that problem. A good hypothesis is one that is testable. Now, sometimes you'll see these written in the form of if something, 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 then something, something, something. You can choose to do that if you'd like. If that's easy for you, if you prefer to do that, please go ahead. I'm not going to require that. That's kind of an older style. All hypotheses predict an outcome. So if we go back to my original salamander question, an example hypothesis might be that the salamanders have curved tails due to a pollutant in the moist soil where they live. Notice that this is a solution to that observation, and I can test it in the next step of my scientific methods. The fun part, designing the experiment. This is what I always enjoyed when I was working in the lab, and I'm sure many of you as well. The purpose of this experiment is to test the hypothesis. When we talk about an experiment, we use the term variable. A variable is a factor in the experiment that is being tested. So this is really what we are testing. A good or valid experiment will only have one variable. So this is really the thing that we change in the experiment. All good scientific experiments follow the following rules. The experimenter makes a special effort to keep other factors constant so that they will not influence the effect of the outcome. Though these factors are called the control variables. So what is the purpose of the control? The controls are not being tested in the experiment. These allow us to compare our results of our experiment too. The other variables that we use in the experiment are the independent variable and the dependent variable. The factor that is changed is what we define as the independent variable, and the factor that we measure or observe is called the dependent. For example, suppose I want to figure out the fastest routes to walk home from school. We will try several different routes and time how long it takes to get home by each one. That's our experiment. Since you are only interested in finding the route that is the fastest, you will do the walking yourself. So you are a constant variable. So let's look at the three variables in our example. The independent variable is the different routes that we take. Route A, route B, route C. The dependent variable is the amount of time it takes us to do that. Notice that this is going to be something that we can measure or observe at the time. And then most importantly, what's our constant? What are we going to keep constant in this experiment? Well, we are going to keep U, ensuring that the same walker is used. It doesn't make sense to have different individuals to do the walking experiment. Imagine one person is able to walk faster than the other person. And therefore, we're not testing the length of the route, but we're testing the speed at which that person or people can walk. One more thing. It is best to make several trials for each independent variable. And this is something, as I mentioned before, we need to be able to reproduce our result. In most experiments, we should repeat at least 3 times before we are confident with the results. So what is a valid experiment? Well, remember that in order for an experiment to be valid, it needs 2 groups, the control group and the experimental group. And always remember there should only be one variable that you're testing. So next is the data. These are the results from the experiment. They may be quantitative, which are numbers, or qualitative, which are things that we observe. So say color. We need to organize them, and we can organize them into charts, tables, and graphs. Next is the conclusion. This is the answer to the hypothesis based upon the data we observed from the experiment. This really sums everything up. As I mentioned, retest. We test and test and test. This ensures that our observations are in fact reproducible and therefore are true. So, in summary, to solve our problem, we identify it, we state our observation, we form a hypothesis, sometimes using an if or then statement. We design our experiment to test the hypothesis, we collect data, we analyze, we make a conclusion, and we retest.